Welcome to Module 2. Having introduced the fundamentals of exposure monitoring and air sampling, we now turn to biomonitoring, soil and water sampling, and advanced considerations in environmental measurement. This module focuses on biological samples, contamination pathways, and protocols for sampling different media. 2.1 Biomarkers and biological monitoring. Biomonitoring involves assessing human exposure by analyzing biological materials. Biomarkers provide direct evidence of absorption, metabolism, or effects of pollutants. Common sample types include blood, urine, breath, buccal cells, hair, nails, or skin, breast milk, cord blood, and meconium for infants. Factors determining body systems affected include solubility of pollutants, metabolic pathways, and duration of exposure. Examples: lead is often measured in blood. Organic solvents may be detected in breath. Persistent organic pollutants can be traced in fat tissue or breast milk. Regulatory agencies such as OSHA sometimes mandate biomarker monitoring for workers exposed to specific chemicals. 2.2 Food and environmental contamination. Food is a major route of exposure to toxic substances. Contamination may arise from heavy metals in soil absorbed into crops, pesticide residues on vegetables, persistent organic pollutants in fish and animal fat, contaminants introduced during processing or packaging. Sampling and analysis of food is therefore important for exposure assessment. Personal exposure assessments often integrate food monitoring with air, water, and occupational samples. 2.3 Soil sampling and contaminant fate. Soil plays a central role in environmental contamination because it can store pollutants and act as a pathway to plants, groundwater, or the food chain. Soil characteristics such as porosity, organic matter, and moisture affect contaminant fate and transport. Heavy metals may bind strongly to soils, while pesticides may leach into groundwater. Monitoring considerations. Representative sampling is critical. Depth, location, and timing of sampling influence results. Both total and bioavailable contaminant fractions should be considered. Soil samples are often analyzed for metals, organic chemicals, and biological contamination. 2.4 Water sampling and quality monitoring. Water is another critical medium for environmental monitoring. Its quality is governed by physical, chemical, and biological parameters. Physico-chemical characteristics: temperature, pH, turbidity, dissolved oxygen, and conductivity. Chemical contaminants such as nitrates, heavy metals, and pesticides. Microbiological monitoring. Pathogens such as E. coli, Salmonella, and viruses. Total microbial counts versus culturable microbes. Sampling protocols. Field procedures must avoid contamination. Containers should be pre-cleaned and preserved properly. Field equipment may include pumps, samplers, and filtration units. Laboratory methods include dilution and plating for microbial counts, membrane filtration for indicator organisms, chemical analysis using chromatography or spectroscopy. Novel methodologies, such as molecular techniques for microbial detection, are expanding the accuracy and sensitivity of water monitoring. 2.5 Advanced considerations in environmental measurement. A few additional issues influence environmental monitoring. Detection limits, accuracy, and precision are crucial in interpreting results. Results below detection limits must be reported carefully. Affect modifiers such as humidity, temperature, and wind patterns can alter exposure levels. Point versus non-point source pollution is a key distinction in water and air studies. Industrial discharge pipes represent point sources, while agricultural runoff represents non-point sources. Low-cost monitoring techniques are increasingly important in resource-limited settings, such as simple field kits for water testing or smartphone-based air sensors. Module 2 Summary. In this module, we examine biomonitoring and the role of biological samples in exposure assessment. We studied food contamination, soil sampling, and water monitoring protocols. We also considered advanced concepts including detection limits, accuracy, effect modifiers, and low-cost monitoring methods. Course conclusion. We have now completed environmental sampling, control, and measurement. Module 1 introduced the fundamentals of monitoring exposures, pollutant classification, personal versus environmental sampling, sampling systems, calibration, and air sampling for particulates, gases, and vapors. Module 2 explored biomonitoring, food and soil contamination, water sampling protocols, and advanced considerations in accuracy, effect modifiers, and low-cost methods. Together, these two modules provide you with both the theoretical and practical basis for understanding environmental monitoring and measurement. You should now be able to appreciate why monitoring is essential, how to approach sampling design, and how results are applied to protect human health and the environment.